Namaste, myself Shivi Saxena, Faculty of Physiotherapy and Diagnostics, Discipline of Medical Laboratory Technology. My topic is Toxoplasma gondii. History and distribution. Toxoplasma gondii is an obligate intracellular conidiae parasite first described in 1908 by Nicole and Menexo in a small North American rodent called Gonidae. Its importance as a human pathogen was recognized much later when Jengu in 1923 observed the yeast in the retina of a child with hydrocephalus and microphthalmia. The name Toxoplasma is derived from the Greek word toxon, meaning arc or brow, referring to the curved shape of the trophozoite. Toxoplasma is now recognized as the most common protozoan parasite globally, with a wide range of the host spread over 200 species of bird, reptile, and mammals, including humans. Classification and identification. Goindia is a coccidian parasite with cat as the definitive host and warm-blooded animal as intermediate host. It is one of the most important parasite of animal. It belongs to phylum Epicomplexa, class Sporozoa, subclass Coccinidicina, order Immisogonia, and family Toxoplasmidae. Morphology. Gonidae occurs in three forms: trophozoite, tissue cyst, oocyst. The trophozoites and tissue cysts represent stages in asexual multiplication, while the oocyst is formed by sexual reproduction. All three forms occurs in domestic cats and other felines, which are the definitive host and supports both um, asexual multiplication and sexual multiplication. Only the asexual form trophozoites and tissue cysts are present in other animals, including human and birds, which are the intermediate host. All the three forms are infection to men. First, trophozoites. The trophozoites is consistent shaped with one end pointed and the other end rounded. The measure 3 to 7 mu in length. The nucleus is ovoid and is situated at the blunt end of the parasite. Electron microscopy reveals an apical complex at the pointed end. The trophozoite stain will with Giemsa stain the cytoplasm appearing azure blue and the nucleus red. During acute infection, the proliferating trophozoites within host cell may appear rounded and enclosed by the host cell membrane. This is called pseudocyst or colony and can be differentiated from tissue cyst by staining reaction. The rapidly proliferating trophozoites is in acute infection or called Trophozoites and trophozoites are susceptible to drying, freezing, and gastric digestion. Tissue cysts. Tissue cysts are the resting form of the parasite. They are found during chronic stage of the infection and can be found in the brain, most common site and skeletal muscles and various other origin. The cyst wall is eosinophilic and stained with silver, in contrast to the pseudocyst. With periodic acid strip P pass stain, the cyst wall stains weakly and the parasite inside are stained deeply. The slowly multiplying parasites within the cyst are called bradyzoites. The cyst is round or oval, 10 to 20 mm in size, and contains numerous cysts. Cysts remain viable in tissues for several years. Oocysts. Oocysts develop only in a definitive host in the intestine of cats and other felines, but not in human. It in oval in shape, in measure 10 to 12 mm in diameter. Each cyst is surrounded by a thick resistant wall. The oocyst is formed the sexual reproduction, gametogamy. And cats shed millions of oocysts per day in fecus for about 2 weeks during the primary infection. The freshly passed oocyst is not infections. They undergo sporulation in the soil with formation of two sporocysts, each containing 4 sporozoites. The sporulated oocyst is infective. Oocyst is very resistant to environmental conditions and can remain infected in soil for about an years. When the infective oocyst is ingested, by it releases sporozoites in the intestine, which initiates infection. Life cycle. Gonidia completes its life cycles in two hosts, definitive host cat and other, in which both sexual and asexual cycle takes place. Intermediate host man or other mammals, in which only the asexual cycle take place. Gonidia has two types of cycle: enteric cycle and exoenteric cycle. Enteric cycle. Anticycle occurs in a cat and other definitive hosts, both sexual and reproduction, gametogony and sexual. Reproduction occur within mucosal epithelial cells of the small intestine of the cat. And cat acquire infection by ingestion of tissue cyst in the meat of rats and other animals, or by ingestion of oocyst passed in its fecus. The Trophozoites are released um in the small intestine and they undergo asexual multiplication, leading formation of merozoites. Some merozoites enter extraintestinal tissue, resulting in the formation of tissue cyst in other organ of the body. Other merozoites transform into male and female gametocytes and sexual sites, cycle gametogony being with the formation in microgametes and macrogametes. A macrogamete is fertilized by motile or microgametes, resulting in the formation of an oocyst, which passes through maturation stage sporulation in the soil after being excreted from host through fecus. A mature oocyst containing 8 sporozoites is the infective form, which may be ingested by rat or other mammals to repeat the cycle. An exoantic cycle occurs in human, mice, rats, sheep, cattle, pigs, and birds, which are the intermediate hosts. Human acquire infection after eating uncooked infected food, meat, particularly lamb and pork, containing tissue cysts. Ingestion of mature oocysts through food, water, or finger contaminated with cat fecals directly or indirectly. Infection from mother to fecus, congenital toxoplasmosis. Blood transfusion and transplantation from infected donor, and sporozoites from the oocyst and bradyzoites from the tissue cyst enter into the intestinal mucosa and multiply. Asexual and trichozoites are found endodigony. Pathogenicity and clinical features. The outcome of toxoplasm infection depends on the immune status of the infected person. Active progression of infection is more likely in immunocompromised individual. Toxoplasmosis as in acquired great importance as one of the major fatal complication is in acquired immunodeficiency syndrome, AIDS. Most human infection and asymptomatic. Clinical toxoplasm may be congenital or acquired. Laboratory diagnosis. Firstly, microscopy, serodiagnosis, molecular and imaging, and other. Microscopic trichozoites and tissues detect in blood, sputum, and bone marrow. Aspirates, stain use GIMSA, PAS, and GMS. Serodiagnosis, antibody detection. Firstly, test for detecting IgG antibody, ELISA, IFAT, latex phagocytation test, and 7 Friedemann dye test. Second, test for detecting IgM antibody, double sandwich IgM ELISA and IgM ISAGA, and test for detecting IgA antibody, double sandwich IgA ELISA, and antigen detection by ELISA. And molecular diagnosis, PCR, imaging, MRI and CT scan for central nervous system involvement, and USG for congenital toxoplasmosis, and other animal inoculation and skin test for Frankel. Treatment, congenital toxoplasmosis. Neonates with congenital infection are treated with oral pyrimethamine 1 mg per kilogram daily and sulfadiazine 100 mg per kg with folinic acid for 1 year. Systemic corticosteroid may be added to reduce choroiditis, and sulfamethoxazole is the drug of choice. Is cannot be tolerated by patient and Depons pyrimethamine is the recommended alternative drug of choice. Thank you.